Hello, everyone. My name is Islam Muhammad Saad Din. It's my great pleasure and honor to receive the IETS Foundation Early Career Achievement Award for 2022. And I'd like to thank all the organizing committee for the IETS conference this year. I will give a presentation about my career and research and experimental results in the last 10 years. I was graduated from Zagazig University as a DVM in 2003. Then I worked as an instructor and teaching assistant from 2004 up to 2007. Then I traveled to Niigata University and worked as visiting scholar and learned the principle of molecular biology and proteomics. Then I joined Seoul National University as a PhD student and I got my PhD in 2012. Then I continued my work in Seoul National University in 2013 and 2014 as a postdoc. Then I traveled to King Saud University and worked as associate professor until 2021. Currently, I am working as a research professor at Cheongnam National University. Started in 2021 August, and currently working as a research professor in this university. I'd like to start my presentation by the words from Mr. Holmes. Mr. Holmes said that if man's mind once stretched by a new idea, it never regains its original dimensions. So this is to acknowledge what are the values of the new idea and what I make during my research in PhD and postdoc. I will talk about the main points of my research is trophoblasts and exosomes or extracellular vesicles, and how to use the somatic cells instead of sperms to obtain blastocysts, and some studies about the thermotolerance of oocyte and communal cells. First, this research about trophoblasts. I was so lucky to attend a lecture from Dr. Fuller Basil when he was a visiting professor at Seoul National University in 2010. And I learned from him what is the meaning of trophoblast and what are the factors secreted by trophoblast, especially interferon tau. Then I developed an idea how to use this trophoblast as a donor cell for nuclear transfer and cloning, and how to study the trophoblast as a model for examining the transgenesis efficiency. Then I applied the ideas for pigs and camels. And also thankful to Dr. Neil Talbot for his outstanding research for trophoblasts isolation and culture. First, trophoblasts are known as the first cells to differentiate from the fertilized egg, and it's considered as oligopotent stem cell and give rise to all kinds of placenta cells to maintain the stages of implantation until the parturition or delivery. And in the early stages of embryo, especially bovine embryos, trophoblasts secrete interferon tau and prolactin-like peptides or signals. And interferon tau is essential for maintaining or establishing the implantation and pregnancy and maternal recognition of pregnancy in cattle, because it stops the signals for oxytocin receptors and decrease the or stop the signals of prostaglandin F2 alpha and maintaining the corpus luteum function. So first experiment, I compared the blastocyst cultured in defined medium or undefined medium. Everybody knows that there is synthetic oviductal fluid medium, and it contains a PSA, but it is like what it's undefined medium. So I cultured the blastocysts in defined medium developed by Seoul National University and Seoul Milk Company. And this medium contains BVA and glucose and myoinositol, and it's two-step culture medium. For 5 days, in one medium, and then moved the blastocyst from the day 5 to day 7 or day 8 in another component of the medium. So in this study, I studied the different culture medium on the temporal expression of interferon tau and associated genes like CDX2 and OCT4, NANONG, and GATA2 and GATA3. So we found that if the blastocyst cultured in a defined medium, it has a smooth and stable interferon tau expression and directly proportional increase in the interferon tau, together with decreasing in OCT4 expression, with a stable CDX2 and ETS2, that's the major components and major factor controlling the interferon tau expression. So we recommended to use a defined culture medium, 2 stages defined culture medium, as a good option for culturing the bovine blastocyst. In second, I cultured these trophoblasts from the blastocyst, and I got nice passages until the passage number 6. And I compared the trophoblast with adult fibroblast as a donor cell for nuclear transfer. And I got this model from Dr. Tsunoda and Dr. Kato from Japan, and they also considered the trophooctoderm nucleus as a donor cell for nuclear reprogramming or developing totipotency after nuclear transfer. So we found that if we use a trophoblast and compared with adult fibroblast and the normal IVF embryos, interferon tau has very nice expression and proportional increase in interferon tau in compared to the cloned embryos using the adult fibroblast. So we also consider that there is an epigenetic memory after using the trophoblast with high expression of interferon tau. Is this expression of interferon tau maintained in the trophoblast and is considered as an epigenetic memory? But this research needs more confirmation and more detailed results. Then we used the trophoblast as a donor cell for nuclear transfer after modifying or genetically modifying these cells. So we got transgenic blastocyst and cultured the trophoblast over the feeder cells and used these donor cells for nuclear transfer, and we nicely got transgenic blastocyst from trophoblast as a donor cell. And also we use the trophoblasts as a model for judging the inducible piggyback transposition and transgenesis. The benefit from using inducible gene expression is to decrease the harm of stable transgene expression. If the transgene is stably expressed in the cells, it can affect the cellular physiology in some cases. So it's good to use temporal and conditioned gene expression as we develop this tool to make a TET-on or tetracycline dependent or doxycycline dependent expression. So we developed this vector and transfect the cells with Fusion HD and select the transgenic cells by neomycin for 14 days. Then we developed the cell line inducible by doxycycline and used for nuclear transfer. After that, we got trophoblast culture from these transgenic cells and applied this model for trophoblast culture. And after adding the doxycycline, the RFP signal increases. When we remove the doxycycline from the culture medium, the RFP signal it also abolished or decreased. So we developed like switch on, switch off model using the trophoblast culture, and it is very good to judge the transgenesis before transfer these embryos to develop for 9 months in pregnancy in cattle. So this model as a switch on and switch off, and very nice model. And we developed a patent of invention from this study. And then I moved to work on blastocyst and trophoblast culture in porcine. And I used some ideas how to develop a good culture method for trophoblast in porcine. So first, I used blastomeres aggregation. So here we removed the zona pellucida by Brown's enzyme and cultured for BZM5 medium. And after 5 days, this aggregated blastomeres contained blastocele and also developed blastocyst. And if you can see, the size of the blastocyst from the aggregated blastomeres is almost 5 or 6 times more than the normal conventional embryos. So we use this big size or giant blastocyst for developing a trophoblast culture, even without feeder cells, like using matrigel or feeder-free culture condition. So we developed culture and condition for for porcine trophoblast culture. Another method to improve the trophoblast culture from porcine, I developed a post-maturation zona perforation. The idea is very simple, is to perforate the oocyte after maturation and used as a good model for blastocyst expansion and blastocyst hatching, and compare this with the conventional parthenogenesis. And in the day number 6, just the day number 6 zona perforation. So we found that day 0 or the post-maturation zona perforation is most suitable for getting good blastocyst number, and hatching efficiency is more increased in day 0 post-maturation zona perforation. And we nicely got trophoblast culture until the passage number 10. Then when I moved to Saudi Arabia, also I have the same idea how to culture the trophoblast from camels, because I worked in camel embryos from 2016 until 2021. So we have the first trial to isolate the camel trophoblast and camel embryonic stem cells. We got blastocyst from in vivo fertilized camels. Then we cultured these embryos until day 21, and this very good record to reach day 21 in culture in vitro. And this blastocyst reached, I think, more than 0.5 centimeters. It was very big size and expansion too much in the dish. And after that, we found that trophoblast starts to extend, to outgrow, and we isolated the trophoblast. And surprisingly, it was very similar morphology to the big trophoblast. So this also confirms that big implantation and the camel implantation are similar to each other. And even when we searched about the placenta type, they are both simple placentation type. So this one, this idea also needs more comparison and comparative study between the pig and the camel trophoblast or placentation. Also, we developed and isolated the embryonic stem cell-like cells from these embryos. We cut the embryonic inner cell mass, and we developed the inner embryonic stem cell-like cells from these embryos. And this was also the first trial to isolate the embryonic stem cells from the camels. Then I moved to study how the cumulus cells from camels are considered as multipotent stem cells. Actually, everyone works with camel or llama or alpaca IVM. We found very hard jobs to culture the oocyte during in vitro maturation. During in vitro maturation, the cumulus cells attach to the culture dish. I never see in the porcine or bovine during my work. So it was nicely to examine what are the characters or what are the properties of cumulus cells. And when we, I'm sorry, uh, when we got these cumulus cells and cultured singly or individually, it developed like embryoid bodies similar to embryonic stem cells. And we cultured this embryoid body. We found there is some expression of OCT4 and SOX2 and KLF and CMEC. They are known that they are the pluripotency factors. So these cumulus cells can differentiate into adipocyte and also can differentiate into osteoblast and into neuron-like. So we confirmed that the cumulus cells are considered as an oligopotent stem cells. Then I moved to an important milestone in my research is to discover the exosomes released from the embryos. Everyone knows that the embryos cultured together in a group culture, even using the well of the well system developed by Dr. Vashta, or zona-free culture together. And even my study to culture the different groups of embryos, different kind of embryos together during using the transwell dishes, transwell inserts, we found that if we cultured the parthenogenetic embryos with nuclear transfer embryos, this developed more blastocyst. So it means a group culture of embryos are essential to get more blastocyst. But what are the signals that can uh, enhance the blastocyst formation? So I also was very lucky. I attended a lecture from Dr. Vatajak. He also visited Seoul National University in 2013, and I was a postdoc at that time. So he just gave a hint about what is the exosome, what are the exosomes. So I thought about this idea during my work for group culturing of embryos. So why don't we study the exosomes released from the embryos? So there was also a question: is exosomes can enter or can penetrate the zona pellucida? And also I found this answer of question from Dr. Anne Van Sum, because she worked very nicely in viruses and how can viruses enter the zona pellucida during her work in RFD and several works for viral infection to the in vitro produced embryos. So I collected the medium from parthenogenetic embryos and cloned embryos, and we firstly isolated the exosomes. At the time, we named at exosomes, but currently there are some regulations from the International Society of Embryo for extracellular vesicles, and they said that these structures are now considered as extracellular vesicles, not exosomes or microvesicles. So we isolated these exosomes and examined some transcripts or mRNA expression for like OCT4, SOX2, KLF, and CMEC and NANOG, and we found most of them are expressed in these exosomes, and these exosomes can be penetrated and located in the developing embryos, and you can see this fluorescent stain exosomes by stain BKH67. And we developed the idea. There is some communication between the embryos, and even this communication can be applied for the endometrium and make embryonic maternal crosstalk. And actually, this was the first research that we published, and we wrote a very nice review. And these two articles are now cited more than 200 times. Then when I moved to Seoul National University, I also worked on oviduct exosomes from the dog model. And everybody knows that the oocyte maturation in dogs occurs in the oviducts, not in the Graafian follicle. So we studied how the oviduct exosomes can affect the oocyte maturation in dogs. And we found that by proteomics, there are so many proteins that can affect the MAPK signaling and the cumulus expansion or oocyte maturation. And these exosomes can also affect the sperm penetration and fertilization. Again, we continued work with oviduct exosomes, but in this time with porcine model, and we compared what are the optimal conditions to use oviduct exosomes for parthenogenetic activation and for somatic cell nuclear transfer embryos. And we found that oviduct exosomes enhance the blastocyst formation in parthenogenetic embryo and also cloned embryos. And if we inject these exosomes in the perivitelline space by microinjection, it also increases the blastocyst formation. And we found that the blastocyst reduces the reactive oxygen species and oxidative stress, and also reduces apoptosis and increases lipid metabolism in the blastocyst for the porcine. Lastly, we worked on shuttle transfers of some mRNA transcripts from male to female cells, but in this case in the rabbit model. Also, as you might know, that rabbit is an induced ovulator animal. So the ovulation only occurs after insemination and mating between males and females. So there are some factors and signals like nerve growth factor. Many researchers said that nerve growth factors is the main factor that induce the ovulation in rabbits. But we have an idea. What if we studied other factors like mRNA in the male cells, like prostate cells and testicular cells and epididymis, and to isolate the exosomes and analyze what are the contents of these exosomes. And we proposed a model that there are some factors secreted from male and worked on the female, especially GnRH1 and kisspeptin and midkine and neurotrophin and NGF2. And also we studied the ADAM17, or this enzyme that can enhance the cumulus expansion. So we developed this idea, and this research is under review, and it's presented in this conference by abstract number 112. If you want more results in the abstracts, you can find this in this result. Also, I have an unusual idea how to use the somatic cells instead of sperm for getting blastocyst. The idea was crazy or somewhat unusual, is to inject a somatic cell in a premature oocyte and putting the oocyte in vitro maturation for 44 hours, because we worked in porcine. Then we found that the oocyte contains two polar bodies, and there is like reduction in the chromosomes for the somatic cells. And when we activate this oocyte, like normally with parthenogenic activation, we developed transgenic cells like RFP cells, RFP expression in the embryo. I'm sorry. RFP expression was in the two cells and morula and even in the blastocyst. But when we make a karyotyping, the aneuploidy was increased up to 80%. So this method needs more improvement, and especially for cell cycle synchronization. What if we played in the cell cycle of the injected cells, and to develop this idea to get blastocyst even in sterile patients, just to take the somatic cells from the skin or from any place in the sterile patients, so we can use these somatic cells to fertilize the oocyte. Then an unusual idea, and this happened by mistake from the person who appointed to get the ovaries from the slaughterhouse. He put very hot saline in the thermos. So when I received the ovary and I checked the temperature, I found the temperature it's around 50 or 51. So I was surprised, and I can see I can feel how hot is the ovary. So I insisted how to culture these ovaries and to culture the oocytes and to establish an experimental model to study the thermotolerance of the camel oocyte and camel somatic cells. Surprisingly, we found that if we culture the oocyte of camels for 45 degree for 2 hours or 20 hours, the oocytes are more sensitive to high temperature. So meiosis is very sensitive to temperature. While the somatic cells, we found some morphological changes. The cells change it to circular shape or spherical shape. Then after recovery in 38 degree, the cells regained the spindle morphology and it can regain the mitosis and it can reach the confluency. And we make like scratch assay or wound healing assay. So we expose the cells and just make the scratch and wound healing. So after around 7 or 8 days, the cells were regained its size and replication, and even the wound was healed. So we investigated this research deeply, and we studied the proteomics, and we found that there is a heat shock protein 1, or similar to the heat shock protein 70. It was very increasingly expressed in the cells at recovery stage. So this uh, thermotolerance of the camel cells we saw in the time-lapse video. As you can see in this video, these cells are normal cells. When we increase the temperature to 45 degrees, the cells become spherical and rounded. And at this stage, we regain the temperature to 38, and some cells regain the spindle shape and even motility. You can find everywhere the cells regained motility and replication until the day number 9 or day number 10. The cells become confluent, and it's similar to the normal cells. So currently, I'm working on developing an engineered exosome through CRISPR-Cas9, and these exosomes will be used for improving the implantation and cloned and transgenic embryos. And also, I am curious about the oocyte intraooplasmic vesicles. We studied the extracellular vesicles, but we have a very big size cell. It's like it is oocyte, and it contains so many vesicles inside the oocyte. So we are now studying what are the intraooplasmic vesicles. Finally, I'd like to acknowledge the IETS Foundation for this prestigious prize. And also I'd like to thank my colleagues and my supervisors in Zagazig University and in Seoul National University and in King Saud University. And my deep thanks to the nominators that recommended my application to IETS Foundation, Dr. Wani and Dr. Anne Van Soom and Dr. Pierre Komzoli and Dr. Jonggi Cho. And finally, I'd like to thank my family for their patience during these hard times in my study, my wife and my daughter and my 3 sons. And also I'd like to thank my current fund in Korea. And thank you for your attention. And if you can contact me, you can, here is the contact, like my email address, and hopefully we can meet in the next meeting next year. Thank you so much.